Hi, David.
Hi, Antonio. How are you?
Fine. And you?
Fine, fine. Just busy, and the weather is horrible. It was nice, but now it's cold, cold. We have 10 degrees or something. It's ugly, but
Oh,
it's fine.
okay. Do you have already booked for Sardinia the flights?
Not really, because we are coming by car.
Ah, okay, okay, okay. So
But I just checked, to be honest, I just checked the ferries. And for me, the easiest is to come to Olbia 20th in the morning
yes.
and to continue to Napoli on 30th, which then boat goes in the evening from Cagliari to Napoli.
Yeah.
So this is roughly, so from 20s to 30s seem to be for us doable without any issues. And it perfectly fits my plans in Napoli because we are supposed to start the next week after the first week of June working in Napoli with
Okay.
Vincenzo. So if this is fine for you, and then I want to discuss some details, but probably later when I can send some more questions because we will have car with tent, so we can even do some camping. So I'll discuss with you.
Okay. Yeah. For this, maybe we will have a call
We can have a call.
when
Exactly.
you want. Yes.
Do you have any preferences? What about Friday?
No, no, no. For me, is the same. Just sometimes is cheaper during a week. Check to if you can travel
Yeah,
during.
I'll try, but for me, it's honestly we have a bit tight schedule.
Okay.
I'm afraid that we don't have much flexibility, but I will check. I'll check. But anyway, do you think we can call each other this Friday? How is
Ah,
your?
yes, yes, yes. Just send me.
Okay,
Yeah.
I'll remind you in the morning, and we find some good time for that.
Okay, perfect.
Fine.
Okay. So let's start this
Yeah,
lesson.
I'm ready.
I'm just here to introduce Professor David Modry. For those that works with me, they already know. And for students of David, there are students of wildlife management and
Mhm.
also PhD students of veterinary medicine course. And so um, David is a researcher that works in parasitology, but also in infectious disease, and works at Brno faculty in Czech Republic. um, Today we will focus to one of his best research topics, and I'm pretty sure that you will enjoy. And so I don't know if I will stay for a long time, but if I don't, please
No
open
worries.
microphone, ask. Otherwise, David, just close the lesson when you finish. Okay.
Okay, super.
Thank you
Fine,
again. Also, on behalf of Professor Scala.
fine. So thank you very much for invitation. And it's one of my favorite topics, so I'm honestly very pleased that I can talk about these nice worms to you. So I will share the PowerPoint. Hopefully, where is it? I never know. Here. Just confirm me that you see the slide.
Yeah, perfect.
And is it moving?
Yes.
Okay, super. So today I'm going to speak about Angiostrongylus, which is quite spectacular, I would say, nematode or genus of nematodes, because it includes several species of more than average importance. You as students or colleagues involved in veterinary medicine more probably know Angiostrongylus vasorum, but vasorum is only a little part of my talk. I will almost avoid speaking about vasorum because today I want to show you mainly other other species of the genus. Yep. So all these nematodes we can group within the huge suborder Metastrongyloidea, and it's quite an assemblage of different genera of worms. Some of them are of major importance. Many of them you probably heard about. Some of them are of, I would say, lesser importance and so on. So it's quite a complex group of taxa, and we don't go today into the taxonomy, I promise. We speak about biology. Most of these worms, with some exceptions, they have quite complicated life cycle. Here in blue, you see the red, but basically it stands for all mammals. I will speak later about reds. So wherever is the blue, something with red, it means in many situations mammals in general. So this is the definitive host, having the parasites usually somewhere in the circulatory system. It can be in the commonly in blood vessels, in arteries. Then there is obligatory intermediate host, which is most typically a snail or slug, let's say mollusk. And in many of them, there are parathenic hosts, which are, we tend to consider parathenic hosts something a little bit less important than, for example, intermediate hosts. But in the epidemiology of human and animal infections by Angiostrongylus, parathenic hosts commonly play a crucial role. I'll show you. So this is a bit chaotic table, but I just wanted to say in that there are different types of the transmission with different types of intermediate hosts, but the absolute majority of Metastrongyloids has indirect life cycle, where the intermediate hosts are the mollusks, and parathenic hosts can be or cannot be involved. The most typical example of the simplest version of the life cycle, which is probably the ancestral one, this is probably the life cycle, the original life cycle of Metastrongyloidia. So the most typical examples of these are those of herbivores. Of course, the herbivores, they don't hunt other animals, so they can barely be infected by parathenic host. So they are simply infected either by accidental consumption of the mollusk, or theoretically also by the larvae, which are being released from the mollusks under some circumstances. We don't know much about that, but most of my talk today is about Angiostrongylus cantonensis, the specific worm. And because it's ideal laboratory model, I do believe that we can learn a lot about other metastrongyloids, just examining properly the biology of Angiostrongylus cantonensis. So many aspects that I will discuss today probably correspond also to unknown features of other metastrongyloids. Yeah. So in most of them, this is the snail, the intermediate host. Down there is a note that in the genus Metastrongylos, those are the earthworms that play a role of intermediate host. Yeah, Metastrongylos is in suids, in domestic pigs, in wild boars, and so on. So not only the mollusks, but in majority of them, it's a mollusk, the green circle you see. Then if we move more towards carnivores, I'll go to that later in the table. There is typical inclusion of the parathenic hosts. Why? Because not that many carnivores eat the mollusks, to be honest. The snails and slugs, they are not very tasteful. If I am a cat or dog or skunk or whatever is here on the picture, I would definitely prefer eating some parathenic hosts than eating ugly snails. And this is what's really happening. So this parathenic hosts in the life cycle on the right, they are symbolized by mouse, is infected by ingestion of the intermediate host, and then the carnivore is infected by ingestion of the parathenic host. So this is the most universal type of the life cycle of majority of Metastrongyloidia. Then we have few species, my table is a bit messy, but then we have few species which exceptionally have direct life cycle, probably secondarily simplified, which are the genera Philaroides, Ostlerus, and Halocercus. You probably heard about some of them, but I'm not going to talk that much about this. So just to keep in mind that there is simplified version of the life cycle, even about the Metastrongyloidea. And then there are other, which are typical for the sea mammals, which cycles through the fish. We don't know much about their life cycles, but these are primarily fish eaters, like the cetaceans or some pinnipeds. And they got infected by eating the fish, which is in the position of intermediate host, not the parathenic host. So it was just the introduction to the whole Metastrongyloid. When we go, or when we dive more deeply into the genus Angiostrongylus, which is the topic of my today's talk, we can say that it's a not very big, not very small. There is about more than 20 species involved in the genus. Many of them are almost unknown. Yeah, some of them were just described once and they've been never seen since, and we know nothing about them, like Angiostrongylus petrovi in Dormouse in Azerbaijan. Probably nobody saw it again. But many other are quite important, and for several of them, we just learn about their importance because we are just discovering these things. Anyway, I'm not going to speak about all of them, no worries. So these are the species that I consider more or less important. And here they are grouped based on the host and site of the infection. So there are some which are typically in mesenteric arteries of rodents, which is costaricensis, well-known species. I'll speak about that. Then Schmidti and Siamensis, they are almost unknown if they are even existing. So this is the first group. Then in the middle of the slide, we have the largest group, which are nematodes that resides in pulmonary arteries of rodents. Some of them are less known, like Moreai, Schmidti, and Andersoni. So they are almost unknown, but many other, and I'll speak about them today. They are well known and well studied, including Angiostrongylus cantonensis, which is the main species I will speak today about. It's in the second position in the middle column. And then there is a third group, which involves those Angiostrongylus species that primarily live in the pulmonary arteries of carnivores, as adults, I mean, which is typically Angiostrongylus vasorum, the one that everybody knows. But beside that, there is also, sorry, I'll just drink a bit. Beside that, there is also Angiostrongylus chabodi, which is typical for wild cats and being also repeatedly documented in domestic cats. Angiostrongylus daskalovi, which is well known in European badgers, and two more species, Felineus and Raelieti, which are again almost unknown. So this is the spectrum we can consider for our future discussions, some of them less important, some of them more. What makes them really important is that some of them are zoonotic. It's definitely Angiostrongylus costaricensis. It's definitely Angiostrongylus cantonensis. It's definitely Angiostrongylus malaysiensis. Maybe also some other, but if so, we don't know yet about this. So these three are zoonotic, and it's good reason for understanding them a bit more. And what makes them even more important, those in red, which is cantonensis, maceraceae, and malaysiensis, they show very strict neurotropism in their larval development. I will have slides for that separately. So they really affect the brain. And because two of them are zoonotic, it means they infect the brain of humans. And you know, we are, us as humans, we are proud of our brains. We are just monkeys which are proud of their brains. So always when the infection is kind of neurotropic, it gives it like really importance. And this is why we nowadays care a lot about these worms, because they are getting closer and closer to us. They are associated with some synanthropic rodents, and they are neurotropic. And nobody wants to have larval nematodes in his brain or her brain, I guess. So, as I said before, the major topic of today's talk is Angiostrongylus cantonensis, which is a well-known species. I will describe you shortly in the next slides. But from the beginning, I want to say that relatively recently, there was another species discovered, which is called Angiostrongylus malaysiensis. And in many studies, probably these two are not properly recognized or distinguished. So it's well possible that several of the older papers that say that they deal with Angiostrongylus cantonensis, in fact, can relate also to Angiostrongylus malaysiensis, or theoretically even to mixed infection, because it was repeatedly documented by DNA-based studies in past few years, that in some situations, the infection can be mixed. Some mollusks, for example, in Southeast Asia are individually infected by bollworms. So we have a mixture of larvae coming from them. So many of the older studies can be a little bit biased by the fact that it's not clear what the people studied. If you look properly on the picture, you see several worms in there. 1, 2, 3. How many? 1, 2, 3, 4. But what's important, there is only one male. Those of you that are trained in the helminthology, at least a little bit, you see a male there, which is the pale worm somehow in the middle, the one forming kind of cordiform shape in the left lower part, the pale small one, little tiny one, it's a male. If you look a little bit more closely to his end, so the end, which is most on the left side, is ending with a spicule, which is that tubular structure that helps during the copulation. So this is typical male having bursa copulatrix and the spicule. The big ones, they are females. They have very typical appearance and they are quite big. So during the necropsy, I'll show you some picture. It's not that difficult to discover them in the arteries. Yeah, they can be up to 2.5 centimeters, the female. So it's not a tiny worm. It's among the strongylids, it's a significant animal. So we are back to the life cycle. So this is typical life cycle of the species we are now speaking about. And there are several reasons why, I already mentioned them in the beginning, why we care that much about this relatively newly studied, newly in Europe, studied nematode. It's zoonotic one. So in the rule of parathenic hosts, also accidentally infected the humans can enter the life cycle. No, sorry, not in the rule of parathenic host, in the rule of aberrant host. Let's call it aberrant host. But not only humans, also the domestic animals, also wildlife. So this parasite is one of the most prominent examples of necessity of One Health, the paradigm that we need to understand its circulation, because we cannot really separate the human parasitology from domestic animal parasitology and from wildlife parasitology, because in this case, obviously, all is interconnected. So this is one of the good reasons why to care about this particular worm. The aberrant hosts that involve the domestic animals, wildlife, and the humans. Then another good reason is, as I mentioned before, that larvae of this nematode in the definitive host or in the aberrant host, they are strictly neurotropic. So when you get infected, most of the larvae, they almost immediately go to your brain. And they can cause troubles, of course. The other good reason is that the parasite is reaching global distribution. We know it originates somewhere, evolutionary, I mean, it originates somewhere in maybe Southeast Asia. And it was also described in there, in Canton. But since then, the parasite spread across the tropic and subtropic regions of the world, reaching slowly the warmer part of temperate regions, for example, in Mediterranean. I'll speak about it. So it's spreading, another good reason to care. And the last but not least, and it's connected with its spread, it's connected, its life cycle is connected to the invasive hosts that are really having global importance, which are the rats and the mollusks. Both of these groups of animals are really important pests and typical invasive animals. They stand among the most important invasive species of the animals on the planet, just next to humans. But it helps definitely the distribution of the parasite. The worm cannot swim across the ocean, of course, but the rats are crossing the oceans there and back, mainly on the boats. The same the snails, and this is the way how the parasite is being spread, reaching even the very isolated islands around the world. I worked in 2017, it was, I think I worked with Angiostrongylus in one of the really remote islands of French Polynesia. When you are there, you really feel this is the end of the world, but the parasite was there, which means it arrived in the past half century. They say that the real spread of the worm happened after the Second World War, but still it's very recent. So this is more or less, it's not really actual the map, but more or less the distribution of Angiostrongylus cantonensis, which we can use as a background for our discussion. So it originates in Asia. From there, it's not that far to Australia and Southeast Asian islands. From there, probably it spread further down to the New Caledonia, New Zealand, Hawaii, Polynesia, and Hawaii. But the opposite direction, the parasite was also probably relatively recently transported accidentally, of course, to South America, where it's present in several countries. This map is quite old, so nowadays that would be a little bit more red on the map, and also to the United States. There is one thing which is misleading on this map, and it's that the countries are, once the parasite is recorded, the country is fully in red. So for example, in the United States, it's definitely not distributed across all the continent. We know it's present in Florida. We know it's present in Louisiana, I think Georgia, and in some of those very eastern parts of the United States, but it's not reaching the border with Canada, definitely. Not yet, at least. And the same is true for China. It's not distributed up to the Mongolian border with high probability. So it's just in the wetter and more warm parts or warmer parts of China. So this is again the life cycle that shows the ways of the infection of humans. This is another typical feature of Angiostrongylus cantonensis in particular. But in this case, this is exactly what I said, that it can apply also to other species, because there is no doubt that basically even with Angiostrongylus vasorum, the routes of the infection would be the same, at least what we know, and probably with many other Metastrongyloids. So there is the definitive host on the left side, that's clear, shedding the L1 larvae in the feces. So far, all clear. If we want to diagnose them, we should use the typical larvoscopy. Normally, we use Berman. Then the larvae, they undergo the development from L1 to L3 in snails. Also, not that surprising, many Metastrongyloids, they do so. And then the rat can get infected by L3 by ingestion of the snail, for example, and life cycle is closed. This is the rat issue. So far, not that important for humans. But what's important is the right side of the picture. And the humans, they can really get infected different ways. And even one way is probably missing here. No, it's there. So the humans, they can get infected by L3 larvae, either directly by consumption of the snail or by the larvae that are liberated from snails. I'll speak about it later. Then the humans can get infected by vegetables contaminated by the snails, usually in situation when the small snail or slug is accidentally chopped into the vegetable. We can also add to this, it's not mentioned in the picture, but we know it. We can also put there the vegetable or fruit drinks, because there are repeatedly repeatedly reported cases of human infection with high suspicion that the humans got infected by accidentally chopped snail or slug in a juice. You simply put the fruits into the mixer, you mix it, and if there is a small slug and you overlook it, then it's done. But what's important, and repeatedly we speak about that, there is a huge group of so-called parathenic hosts, or we can part of them call true parathenic hosts, and part of them can be considered only short time transport host, which are crustaceans, reptiles, and frogs, which is another source of the human infection. So there are a lot of different ways, and the transmission itself, it depends to humans, I mean, because this is what we care about mostly. It really depends on the local food habits. So basically, it's a parasite that's really related to human culture. I'll show you later. If we look a little bit more towards or close to the Europe, because this is something what's interesting for us, starting from the right side, the infection was described in the 80s in Egypt. Since then, there were not many studies from Egypt, so we don't know exactly how it is. I will tell you in my next lecture in a couple of months or next year, because two of my PhD students are next week just starting the trip to Egypt to search for Angiostrongylus cantonensis. So hopefully we will know more soon. So this is the Egypt. Then if we move to the opposite side of Africa, the infection was discovered more than 10 years ago. It was, I think, 2010, so it's already 15 years ago almost, in the Tenerife island of Canary Islands. And since then, it was reported also very recently on some other islands of Canary Islands. So it's well-established focus of Angiostrongylus cantonensis in rats in there. However, there are no human cases known from there, I have to say, interestingly. And relatively recently, something like 3-4 years ago, the parasite was discovered on Mallorca, which is in the Western Mediterranean, and 2 years ago on Valencia on the coast of Spain. Beside that, there is one anecdotal report of human infection in France of unknown origin. It was in Paris. And that's it. The rest of Mediterranean basin, we don't know. And definitely it's something to search for. And again, I truly hope I will report you soon about at least the presence or absence in Italy, because we are just starting research in Napoli region, in Campania. And so we are going to trap the rats during this spring, and hopefully we see either negative or positive results. At least we will have some insight into the situation. Because the fact that there are no cases known in rodents or even in humans and dogs, honestly, in Eastern Mediterranean, it doesn't say that it's absent there. Maybe it's totally overlooked, and maybe the parasite is sitting in there for ages. We have no idea. So it's something that deserves attention. Just to introduce you a little bit closer to one of the European foci of Angiostrongylus cantonensis. So in the Tenerife, it was originally described from northeast part of the island, from Anaga region, if you know the island. And since then, it's reported across mainly the humid part of the island, which is the green darker line, which basically lies on the northern slope of the volcano. Tenerife, most of the Tenerife is a volcano. So the parasite is mainly in the humid regions, but we already know that it penetrates also deep to the dry south of Tenerife. And one of the major hosts in Tenerife, I mean, the definitive host is clear. It's black rat and brown rat, Rattus rattus, or Rattus norvegicus and Rattus rattus. So they are both common in Canary Islands the same way as they are common in the Mediterranean as well. But the common intermediate host is very specific snail, which is called Plutonia lamarcki, which is this one. And it's wherever we studied this snail in Tenerife, it seems to be more commonly infected than the others. It's probably because of its lifestyle. It's a very active snail that crawls long distances, actively search for the food, and also live in humid areas where the transmission is easier. So we can conclude basically that all the mollusks, all species of mollusks, slugs, snails, aquatic, terrestrial, then in some or under some circumstances, they can be intermediate hosts. But how frequently they are infected, it depends always locally on the snail ecology, on the rat ecology, and on the environment. So Plutonia lamarcki on the Tenerife is one of the really good hosts. So if you want this worm, if you search for Plutonia, the prevalences are really high. What's interesting, this is a study we did with Jan Slapeta a couple of years ago, when we tried to study the mitochondrial genes of to see the intraspecific diversity of Angiostrongylus cantonensis. So all these clades, down there sits Angiostrongylus malaysiensis by the dark triangle, you see it. And all these groups which are marked by 2, 1, 4, 3, 5, these are so-called haplotypes of Angiostrongylus cantonensis. And what's interesting, that all the invasive lineages we have a chance to study, which was from Japan, Brazil, Australia, Hawaii, Polynesia, and Tenerife, are almost identical. So because of some reasons which we don't understand fully, this is the successful strain or successful haplotype in colonizing the world. The fact that it originates from Southeast Asia is clear, because all these worms originate somehow deeply in their evolutionary history, or not that deeply, in their recent evolutionary history, they originate from Asia. But this one, because of some reasons, started the trip around the world. And this is the one which is also in Mediterranean. It was not known by in that time, but now we know that haplotype 2 is the one which is on Mallorca, and it's also the one which is in Valencia. We don't know so far what haplotype is in Egypt. You can guess that the same one. Okay, so let's go a little bit to the life cycle to see how it works in reality. The definitive hosts are the rats. The rats mean rats, not mice, not other rodents, the rats. Even though probably some other can act as the life cycle in the life cycle, the dominant are those three species in the first line: black rat, or sometimes called ship rat, Rattus rattus; brown rat or Norwegian rat, Rattus norvegicus; and Rattus exulans. I'm not sure about the English name, but it's the species distributed mainly across the islands of Southeast Asia, down to Hawaii and Polynesia and so on. So it's Rattus exulans. So those are the dominant species. The remaining ones, they can act as hosts under some circumstances. So, and this is the life cycle in a rodent. This is the timing and the migration of the larvae and its timing in typical infection of a rodent definitive host. So the rat is infected by RSV larvae, probably usually from a snail. And as you see, in 17 hours, most of the larvae migrate to the brain. So it's very fast, acute infection. Then the larvae are still microscopic in that time. They migrate a little bit through the brain tissues and finally ending up on the surface of the brain, just under the meninge. And here they mold from L4 to L5. In that time, they are reaching significant size, up to almost 1.5 centimeter. And if the host survives this, and if the larvae survive, roughly after 3 weeks or 3 to 4 weeks later, they migrate to lungs, where they reach the pulmonary arteries and they reach the maturity. They copulate and start producing the larvae or the eggs. The eggs, they hatch in the lung parenchyma and the larvae are being released, caught up, swallowed, and shed in the feces. So the whole prepatent period is between 42 and 45 days. Not long, not very short. It simply takes time to develop in the rat. And as I said, the first part, which means the migration to the brain, is happening not only in rodent definitive host, but it's happening also on aberrant host, and we speak about it later. And this is the part responsible for the clinical science. And we can conclude that it's not only in aberrant host, even in rats, this is responsible for some clinical science. And if you have experimental rat, and if you feed it with more than 100 larvae, the rat is dying. It cannot handle the massive infection of the brain. And 100 larvae, it's not much. You can imagine that in one snail of medium size, we can easily have several thousands of larvae. And the extremes say, the colleagues from Hawaii told me that they found a snail, Levicaulis alta, which is quite big, not snail, slug. It's quite a big slug that was infected by more than 1 million of larvae. And if you imagine that 100 larvae can kill a rat, and we have a snail having 1 million of larvae, then apparently the possibility to contaminate some even environment when such snail dies is quite high. This is the brain of experimental rat, and on the, I'm not sure if you see it, but on the slightly on the left side you see the coiled worms, but also here, so on the surface of the hemispheres. So these are the L5 that are just finishing the development before they start migration to the lungs and heart. And these are the adults in the lungs. So you see these are significant worms. If you compare that to one of the lobes of lungs, the size is quite big. And if all goes well, after the prepatent period, the rat starts shedding the larvae in the feces that appear as L1s mobile larvae detectable mainly by larvoscopy in the fecal samples with having characteristic caudal end. Then the larvae are eaten by another intermediate host and the life cycle starts again. In this case, we have to say that a lot of mollusks, both the slugs and snails, are quite keen to eat the feces of mammals. And this is probably the way how they got infected. The active foraging on mammalian feces. I saw very nice videos by Eric Morgan from Ireland showing with night vision a piece of shit of a dog or fox. It was a fox, I guess, over the night, and it was really obvious how tens of the snails and slugs crawl over during the night over the feces, and finally in the morning it totally disappeared. And this is the way how they got infected. So it's nothing accidental. They really like to eat it. Thanks to the research on Angiostrongylus cantonensis, we know that, and it's probably true for other Metastrongyloids as well, that both aquatic and terrestrial snails and slugs can be important depending on the local conditions. And even in our experiments in the lab, we use both types of the snails. We use the aquatic ones. On the left, it's Pomacea, so-called apple snail, originating from South America, nowadays invasive species across the world in tropics. Then the small one is Biomphalaria glabrata, common laboratory snail because it's a host of schistosoma as well. And if we go to the upper row, on the left side, there is giant African snail, Lysachatina fulica, also very common pet snail in Europe, I'm sure including Italy. Those are those large, giant African snails. So if they are captive, they cannot be infected, but if they live in the wild, they can easily get infected, and they are important hosts, and we also use them in experiments. And on the right side in upper row is Veronicella cubensis, another very important invasive snail species, slug species, distributed across the tropics and very potent and important intermediate host. And this is the outcome of the infection of the intermediate host. This is the L3 larva ready to infect another host. Then we are coming to the parathenic hosts, as I promised before. So far, they were quite overlooked in the life cycle of many Metastrongyloids. And case of Angiostrongylus cantonensis shows us nicely, and also our research we did on Canary Islands shows us that they are really important, or with high probability they are important because they are frequently infected. Yeah. However, their role in the natural life cycle is not that easy to study. First of all, because we cannot hunt or kill that many of them. Second, because the parasite can be diluted in the normal ecosystems. And there are also limited experimental data. Having the quite long research on Angiostrongylus cantonensis on Tenerife and in the laboratory enabled us to study this a little bit more in detail. First of all, because Canary Islands are really nice, I would say, natural laboratory with very low biodiversity, but very low population densities of the vertebrates. And second, because in the lab, we maintain 3 strains of Angiostrongylus directly in experimental infection, so we can do a lot of experiments. And if we go to the Canary Islands, we started to work a couple of years ago. Lucia, a PhD student of mine, worked with Galotia galoti, the endemic lizard, which is extremely common and abundant across some of the Canary Islands, specifically on Tenerife. And this is really omnivorous lizards. They eat almost anything. And we looked for their role, or we asked ourselves, can they serve as parathenic hosts in the life cycle of cantonensis if we know that it's present in rats on the island? And the answer was yes, definitely they can. And we found Angiostrongylus in 25% of liver samples we got from these lizards, which is quite a high prevalence. So how much the rats get infected by eating the lizards is difficult to guess, probably quite frequently. But the lizards can play a role in the transmission of the parasite also to other hosts. So they definitely play a role. And very recently, we just finished an experiment with captive reptiles proving that also in captivity, we are able to infect in that case, geckos by feeding them with snail tissues, with L3 larvae. And the larvae tend really to accumulate in the geckos. But we also proven that if we fed this gecko to a snail, the L3 larvae, they move simply from one peritonic host to the other host and continue surviving in the tissues of a snail, extending the probability or the opportunity for being transmitted to the definitive host. So these are the true paratonic hosts. And then we have in the life cycle of cantonensis are also known so-called, not very well defined, but we call them transient hosts, which are hosts that supports the infection, but only for a short period of time. And this can be some mollusks, but mainly centipedes and crustaceans. So it seems that they are not infected for longer than a couple of days. And very recently, we finished the experiment with fish, and it also shows, yes, if you feed the fish with snails, the larvae are able to survive in the fish somewhere around the intestine or in the muscles for a couple of days. But 5 to 7 days after the infection, the larvae already disappear. And this is probably the same what's happening with crustaceans. But I'll go to crustaceans once again. So if we kind of review what I said, we can say that what's particular in discoveries on Angiostrongylus cantonensis, and what's probably true for many other Metastrongyloids, is that they don't only circulate among these hosts, but they basically flow through the food web, through so-called intermediases, where the L3 larvae can change the host. So beside a circle, which is really a circle from mammal through mollusks, paratonic hosts back to mammal, there is probably kind of lateral flow through which the L3 larvae in the relationship between the predators and their prey are being transported through the food chain. And this is what we documented in our experiment with geckos and snails. Yeah, so there was a snail in the beginning, then the geckos got infected, and after the geckos, the snail got infected. Here we have large lizard because varanid lizards are quite important as well. I will go to it. Another important feature in the life cycle of Angiostrongylus, and it also explains probably a lot of infection by Angiostrongylus vasorum in dogs, is the fact that if the terrestrial mollusks die in the water, so it's drowned in the water, like here in a vial, larvae are massively escaping from its body, surviving further in the water. So this can be different between different types of snails, but it's happening always. And you see how fast it is. Yeah, so principally in Veronicella cubensis, which is the green line, one day after the infection, the significant part of the larvae were released to the water, and Veronicella, and then it went down. In Veronicella sloanei, basically it was not very different. If you look, the maximum was even earlier in 7 hours. And what's important, the pie charts on the right side, it shows us how many larvae remained in the snail and were later, this is experimental, later detected by the digestion. So you see that significant part still remains in the dead snail. This is the orange color. But in the same time, a deal of the larvae really are released to the water. And what's important is that they survive up to 1 month in the water, living somewhere on the ground, simply surviving. Interestingly, if they are not eaten by some other aquatic organisms, which has really huge ecological consequences, because it seems that in the normal, well-functioning aquatic ecosystem, if the snail is falling to the water, the larvae are, of course, immediately released, but then they are eaten by different types of plankton and other organisms. We do nowadays series of experiments. I don't have slides for that, but we do series of experiments with crustaceans, with microscopic planktonic crustaceans, but also with bivalves, the freshwater bivalves, the mollusks. And we've proven in the experiments that these animals are effectively filtrating Angiostrongylus cantonensis from the larvae from the water and basically eating it. So they, instead of being involved in the life cycle, they are removing gradually this larvae from the water, which is of course decreasing the chance of infection of other hosts. So this is the normal ecosystem. But if we go to simplified artificial ecosystems, for example, small water puddles somewhere in the cities, where the normal fauna is missing, so the planktonic predators are missing, then probably the larvae of Angiostrongylus can reach much higher concentrations, which is increasing the risk of the infection later for the humans or other hosts. Yeah, so this aquatic phase of the transmission of Angiostrongylus is still quite enigmatic, but with high probability, the same is happening with the dogs and with the snails that are drowning or slugs in their bowls, the water bowls. Yeah, it's at least I have a dog, I have a garden, and it's not that uncommon that stupid slugs are overnight falling down to the water bowl, drowning themselves, and I find them dead in the morning. We don't have Angiostrongylus vasorum, neither vasorum nor cantonensis in the Czech Republic commonly. We don't have vasorum cantonensis at all, and vasorum is rare. But I can imagine that logically, if the snail or slug is infected, and the larvae are coming out over the night, then in the morning, if the dog drinks the water from such a bowl, it can get infected by thousands of the larvae, which can really lead to massive infection with some clinical importance. So let's look a little bit more to the ecology of human infection. So far we spoke about how it circulates in the nature. It's a zoonosis. So let's ask, okay, how can I get infected? Or what can I do to prevent the infection of myself? Definitely the rodents are important, but not as a direct source of the infection of humans, because the L1 larvae are not infective to humans. The L1 larvae should go first through the snails. But still the rodents are, of course, a key element in the epidemiology, because they are abundant and they are living very commonly close to human settlements. They simply live next to us. And all these three species, which are considered being important hosts of Angiostrongylus cantonensis, they are synanthropic. So they are everywhere, and you confirm the same. In Sardinia, you don't have three of them, you have just two of them. You don't have exulans. We don't have it in Mediterranean. But those two species, both Ratus ratus and Ratus norvegicus, are common all around the warmer parts of Europe, reaching very high population densities in the cities, directly in the houses, next to the houses, but also in the wild nature, like in Tenerife. The island is full of rats. And they are invasive. They were not existing then before. And they are invasive even on the Mediterranean islands. It's well known. So many island ecosystems are basically being shaped by presence of high population of rats. So, but this is not important as a direct source of the infection. What's important are these hosts, which means the hosts that sustain the L3 larvae in their tissues. And then if you look to the map of global distribution of Angiostrongylus cantonensis, we can say that in different areas of the world, different way of infection prevails, which shows what I said before, that the infection itself, it's very tightly connected to the human feeding ecology or food ecology, which is not that surprising because it's a foodborne disease primarily. And foodborne diseases simply depends on what we are eating, how we eat it, how we cook it, how we prepare it, how we buy it, and so on and so on. So if you look a little bit to the symbols, the green dots stands mainly for the areas where raw snail meat or where raw snails are being eaten, which is typically, not surprisingly, Southeast Asia. So then probably great majority of cases really originates from consumption of or preparation of this snails and slugs in the kitchen. Then if we go further, the violet color, or what color is that? is a symbol for a human infection by vegetables being contaminated by snails or slugs, as we mentioned before. And this is typically used to explain the infections, for example, in Caribbean. They don't eat snails, or they say at least we don't eat snails here. It's typical also for the infection of in New Caledonia, for example, and so on. I don't have the spot there, but it's there. Then we have the yellow color, which are the crustaceans. The crustaceans are locally well known. The freshwater, I stress here, freshwater crustaceans, freshwater shrimps. I show you a picture soon. They are commonly eaten on some islands of French Polynesia. They reach quite a big size. They are almost as a crayfish. They can be easily 15 centimeters long. When we search for them at night in the small rivers in the forest, you see them everywhere. And people, they really like to eat them. But what's important, they eat them raw. Because us in Europe, we commonly either freeze because we buy the frozen shrimps, or we boil or barbecue them or whatever, roast them. So we cannot get infected, even if the infection is present in the region. But in French Polynesia, they use very specific coconut-based sauce, which is half fermented. They put the raw shrimps in that, and apparently it's optimum medium for the larvae to go out from the shrimps and to stay alive to get infected, to provide source of infection for humans. Then there is the blue spot, which is on the extreme left of the picture, which is Hawaii, where it was repeatedly suggested that the humans get infected from drinking water because of the snails and slugs falling into the wells, larvae being released from their bodies, and then the humans get infected by the water. It's a little bit hypothetical, but the team on Hawaii, the American researchers on Hawaii, they've proven that the larvae can go through some of the systems of water filtering. So this probably is one of the reasons because Hawaii is known for really high incidence of the infection among humans. And they don't eat snails. They don't eat that many vegetables in there, specifically on the islands where they know that the Angiostrongylus cantonensis is present. They really don't eat that vegetables because they are afraid of that. And they still get infected, so it's probably from the water. And the last, which is the orange spot that you see in Southeast Asia, but also in India, is infection by consumption of parathenic hosts. In many parts of the world, frogs and lizards are eaten. And if you eat them raw, which is happening for medicinal purposes or medicinal reasons, in India, but also in Southeast Asia, the humans get infected. And I'll have pictures for that later. So if we go a little bit through the pictures, through the important source of the infection to humans. So in some parts of China, for example, this invasive originally South American snail, Pomacea truncata, is a common source of the infection. It's already a piece of meat. And if you eat it raw, it can easily contain thousands of larvae, which logically leads to clinical infection. African giant snail, it's another important host, but it's not eaten that much, to be honest. It's eaten only very locally in the world. So it seems it's important in the circulation in the natural ecosystems, but it's not that important, in my opinion, as a source of the human infection. Veronicella cubensis, it's quite a nasty slug that is probably not eaten around the world, but it's the one that can accidentally be chopped in the salads, for example, or juices, because it's quite common around the human settlements. It can contain hundreds of or thousands of larvae in that and so on. And the same way, if we go around the world, we find also some other important mollusks. I already showed you Plutonia in Canary Islands and so on. So the transmission biology of Angiostrongylus cantonensis always depends on the particular situation in a given area. And again, we can conclude that this is with high probability the same for other metastrongyloids. It's for sure the same for Angiostrongylus vasorum. Yeah. Because simply in southern Italy, in England, and in the Czech Republic, the ecology of snails and slugs is different. It's different climate, different species composition, different activity of these hosts. So logically, it impacts also on the transmission ecology of the parasite. Yeah, and cantonensis shows that quite nicely. And what's important, that even very small snails can sustain significant numbers of larvae. Because one can hardly imagine that you eat this snail accidentally because it's half kilo. It's a papaya fruit the snail is sitting on. Yeah, so you cannot basically eat it because it's too big. And you definitely cannot eat it accidentally because you probably realize that you are eating something like half chicken. But if you look at this little guy, which is Subulina octona, a common invasive snail around the tropics and also subtropics, common also in Europe, even in the Canary Islands, I can imagine eating it accidentally with your salad, to be very honest. And even these little guys, they can, at least in experiments, contain hundreds of larvae in their small bodies. And this is probably how the human infection happens, by something very small, which is being chopped in the salad or into the water, or into the fruit juice or something like that. So if we go a little bit to, so those are the snails. If we go to crustaceans, so this is the freshwater shrimp of the genus Macrobrachium. This is the one which is a common host in Polynesia. Because humans simply, they go, they catch, they eat them, or they macerate them in the coconut milk, larvae are coming out. So important locally. If you go to parathenic hosts, there are several studies, or few, but no, a few, but some. There are some studies proving the importance of monitor lizards. And not surprising that monitor lizards are so commonly infected by L3 larvae because they are top predators. These are large lizards, yeah, being 1 up to 2 meters big. So these are robust, massive lizards. And they are usually eaten for medicinal purposes. We did some interviews last summer. We did some interviews in southern India, where the infection by Angiostrongylus resulting from consumption of monitor lizard meat are well known. And the local rural hunters really confirmed, yes, there is a trade with varanids, because people, they want it. They want to eat raw liver because they do believe that it will heal the cancer, it will increase the potency or whatever. So people intentionally eat raw liver of these animals. And liver is exactly the place, we know it also from experiments, where the larvae are accumulated. And if you imagine that such a monitor lizard is eating almost anything, the lizards, the snails, the whatever, and accumulates the larvae, then logically the resulting infection in humans can be quite high. And this is what's really happening. Interestingly enough, the same was proven even very recently, even though it's not published in Australia. My colleague Jan Slapeta, he just wanted to check the local monitor lizards because we know that Cantonensis is present in Australia. It's quite important there. We go to that later. So he just asked for some histological slides from the university collection, the slides of liver of monitor lizards. And in one of the few, he found very nice granulomas with larvae inside, which is with high probability, again, Angiostrongylus cantonensis being present in these hosts. And last but not least, study from China proving the infection of humans by eating raw frogs. Okay, so that was the way of infection of humans. So let's check, or let's discuss how it continues, how it develops, what happens if the humans are infected, or if we are infected. The larvae are migrating to the brain, or most of them, they migrate to the brain, causing so-called eosinophilic meningitis. Usually the disease is abbreviated as AEM, Angiostrongylus caused eosinophilic meningitis. And they say that it's the most common source of eosinophilic meningitis in the tropics in humans. The clinical features of infection in humans are with high probability related to the dose, to the number of larvae that you ingest, which is more or less question of an accident. So you can be lucky enough to eat only few and then nothing happens. This explains relatively high seroprevalences in Southeast Asia without clinical science. But if you are unhappy or unlucky, sorry, not unhappy, unlucky, if you are unlucky enough, you simply accidentally eat a snail that contains thousands of larvae, then you probably get infected with severe meningitis, and there are even some deadly cases. It's quite sad, it's not very frequent, but there are little cases of the infection in humans. And in some particular circumstances, the larvae, they go to the eye, causing ocular form. You see it on the lower picture. There is larva in the lower part. And it's not known yet what is the predictor for migration to the eye. Maybe it's totally accidental. And if you know about something about Angiostrongylus vasorum, we see the same happening also in the dogs. And I saw even the cases, or at least one case in Italy, of Angiostrongylus vasorum, the dog Angiostrongylus in the eye. Yeah. So, as I said, fetal cases are quite rare, but it is causing commonly very long time disability, because it's meningitis. So basically, it's similar to any other meningitis. This one is eosinophilic one because it's caused by worm. So eosinophilia is not surprising, but it's simply meningitis. And also the treatment, honestly, it's treatment of the meningitis. So it's not when we really treat, or when people treat the acute cases of the infection in humans, or also in the dogs. I'll speak about it a bit later. So it's treated as meningitis. So first of all, you should decrease the intracranial pressure. Second, you should decrease the inflammation by corticosteroids. Then you can think or not about giving the anthelmintics. But in the later stage of infection, it doesn't help anyway because the larvae are usually dead by that time because of the immunity reaction. So like giving albendazole later than 2 weeks after hypothetical infection is usually not recommended at all. On the other hand, what's really recommended for treatment of humans in endemic areas, if you are aware of possibility that you are infected, it's really recommended to start albendazole immediately because it can prevent the migration of the larvae to your brain or their further development. So it's quite tricky. There are some nice articles about that recently published. There is ongoing discussion how to treat these cases, and there are even some controversies in that. So how is the impact in other hosts? We know that in some areas of the distribution, mainly on the newly invaded areas, Angiostrongylus cantonensis can be quite important pathogen for different aberrant hosts. Interestingly enough, again, it's very locally different, which probably shows some ecosystem differences, but also maybe the differences in capacity of the veterinarians to detect the disease. For example, in Australia, Angiostrongylus cantonensis infections are on the eastern coast, well known in dogs. It's the most common, one of the more common neurological issues in the dogs. And it's well known by veterinarians, and then it's also relatively efficiently diagnosed. Interestingly, from Southeast Asia, we have 0 cases from the endemic areas, which is probably caused by, I would say, the lack of interest first, because simply if you are not looking for that, you will not diagnose it. And the same is true for wildlife. Interestingly, there are 2 species of wild animals which are well-known hosts of Angiostrongylus cantonensis, or I mean aberrant hosts, so which means they are dying due to infection of Angiostrongylus cantonensis. I'll go to both of them a bit later. Now, the first of them is a so-called tawny frog mouse. This is quite large bird living in Queensland in Australia, or along the eastern coast of Australia. And as you see on the slide, its diet consists mostly of insects, slugs, and worms. So with high probability on different types of intermediate or paratenic or transient host of cantonensis. And it's commonly reported as a bird which is dying or suffering of paresis and paralysis, specifically in young animals. And in Australia, it's considered being a good sentinel species for mapping the distribution of Angiostrongylus cantonensis. We can ask why Australia and why this particle bird? Why, again, we don't see the same, for example, in Southeast Asia. We have also plenty of big birds in Southeast Asia. But we are back to what I said in the what I said about the dogs. Yeah. In Australia, the wildlife medicine and the wildlife rehabilitation is really significantly, or I would say even massively developed. So people, they are aware of the importance of the wildlife and they care. And in the same time, the tawny frog mouse, it's big bird. It's big, simply. It's a massive bird. And you cannot overlook it. If you go through a forest, sorry, or not through forest, I wanted to say through city park, in your morning walk with a dog, and you see a bird, which is really giant, somehow paralyzed on the ground, you do not overlook it. And usually you don't leave it there, of course, because it's still alive. This is relatively long-term paresis and paralysis. So you don't leave it there. So what do? You call the nearest shelter, they come, they pick the bird, and you have the case. If the same happens to a small bird in Europe, no one cares because it's small first. And that's it. Yeah? And if the same happens in Asia, the bird is probably eaten. So again, there is no report, so there are no cases. So this is a combination of the reasons why particularly tawny frog mouse, particularly in Eastern Australia, is a good sentinel host. If you go to more cases on the islands, there is an interesting case reported in one of the Japanese islands on, which one? Amami Oshima Island, which is a small island inhabited by highly endangered endemic red species. And of course, as many other islands, the islands is also being colonized by invasive red species. And through the snails and so, as we know, the life cycle, the local endemic rats get infected, and they are the mortalities described among this rare and endangered endemic rodent. So this is another case of, or example of impact on the local biodiversity. And there is one more case which is closer to Europe, and it's quite interesting. And again, it has ecological background, and it probably has also some cultural background. And this is how the infection was discovered on Mallorca, which is not that far from you in Sardinia. If you swim long enough towards the west, you reach Mallorca. And in Mallorca, our colleagues from the university described the lethal cases or the mortality in Algerian hedgehogs, Atelerix algirus, which is the only species of hedgehog living in Mallorca. And they reported the presence of the Angiostrongylus cantonensis in Mallorca was reported based on the series of cases in this hedgehog species. Again, we can ask why. Why particularly this hedgehog, particularly in Mallorca? It was not clear to me before I arrived to the island. And first of all, Atelerix is very common in there. Second, the infection is quite common in snails. It's an island, and the snails and probably also paratonic hosts represent important food for the hedgehogs. So this is the ecosystem part of the life cycle. And of course, the island is colonized by rats, as any other islands in Mediterranean. But the cultural part of the fact is that the hedgehogs is among the largest wildlife on Mallorca. They don't have much wildlife, to be honest. There is almost nothing living there. It's an island. So the people, they care about hedgehogs, and they really have shelters and wildlife rehabilitation shelters where they bring the hedgehogs, which is similar to some parts of Europe. And if you start bringing the hedgehogs to rehabilitation centers, if you start caring about what's wrong with this hedgehog, then the probability that you discover the infection in this host is much higher, of course. Yeah, so this is very similar explanation to the case of tawny frog mouse in Australia. So if we care enough about these little things around us, we probably it increases the chance of discovering the diseases, which is, in fact, quite interesting part, in my opinion, at least, of wildlife health and wildlife management. And because we commonly tend to perceive wildlife as something, of course, valuable, endangered, deserving the conservation, which I fully agree. But in the same time, wildlife is really important sentinel for infectious diseases. So if we want to understand what's happening in some given ecosystems, and if we care about why the hedgehogs are dying, then we can discover a lot of interesting things, which was exactly the case of Angiostrongylus on Mallorca. Yeah, so that's interesting. And what's interesting even more, because in that when it was published, something like 4 years ago, if I remember well, 2019, yeah, so 4 or 5 years ago, it opened a question if it can have really significant impact on the population of Atelarix on Mallorca. And I was really curious about that. And to answer this question, we started some experimental infections. So we worked with similar hedgehogs. We worked with Atelarix albiventris in our laboratory. We performed a series of infections of hedgehogs by Angiostrongylus cantonensis, the same haplotype which is present in Mallorca. And what appeared is that yes, the hedgehogs, they develop the clinical signs, but only after very high dose of the larvae, which explain why the infection is relatively rare among the hedgehogs on Mallorca, or rarely detected. Because simply only some hedgehogs are unlucky enough to eat a snail which is full of larvae and then they die. That's it, bad luck. And also it gives some hope, of course, to the population of the hedgehogs, because probably not all of them will die because of presence of the nematode. Yeah, so it's very similar. And it also shows us something about the human infections. Yes, you can be sick, or the same about the dog infections, to be honest. Yes, you can get sick. If you eat a larvae, you get sick. But depending on the number of larvae, you either get only a little sick, and you probably even don't go to see the physician and you survive without any issues. Or if you are unlucky, or if you have bad luck, you eat thousands of the larvae, then you might die. And this is something which is very difficult to prove, of course, in the real life. Hedgehogs can help us a little bit in understanding this. And it's very difficult to prevent this, to be honest. And when we speak about the reasons of the infections for humans, I forget to speak about the cases which have absolutely unknown origin. There is, for example, a study, quite not nice, but I would say well-done study from the island of Mayotte, the French island in Indian Ocean, where the colleagues studied a cluster of human cases across the island. And all those cases were babies. I think, if I remember well, between 2 and 4 years old. Many, not many, but several of them died because of the infection, which is quite sad. And the reason of the infection, or the source of the infection, was not discovered. So there are probably ways of infection which are difficult to predict. And somehow, in some circumstances, probably either the wildlife or the humans are confronted with a high number of larvae, and then they develop severe infection. That's just a picture from our experiment, which was with hedgehogs. So, as I said in the beginning, before we go to the remaining species of the genus, Angiostrongylus cantonensis offers a perfect experimental opportunities for the research. Why? Because it's one of the few heteroxenous nematodes that we can complete the life cycle, in which we can complete the life cycle in the laboratory. Why? No, because of both typical hosts are relatively easy laboratory models. We can, of course, keep the laboratory rats. Rats are among the easiest and most commonly used mammalian model hosts, and we can very easily keep the snails. So this is why also the numbers of the studies nowadays, not only our team, but also many others, other researchers around the world, do a lot of work on cantonensis. So it's perfect model for other Metastrongyloids. But still, regardless, the fact that there is maybe 600 studies published, what we have in the database about cantonensis, we don't know much about this important nematode. We don't know real geographic distribution. We even don't know how is it in Europe. We don't know how is it in Sardinia. We don't know how is it in Sicily. We don't know the Greek islands. So we don't know that much. We also don't know whether the genetic diversity that is observed is somehow related to the neurotropism of the larvae or not. We don't know the spectrum of the definitive host. We don't know if the infection, like for example, in the Japanese island, it was shown that the infection really spill over to the local rodents fauna. But is this happening frequently or not? We don't know. We don't know what's the real consequence for wildlife in the newly invaded fossae. We don't know much about fish as parathenic hosts, or I have to say we know already because Lucia, my PhD student, she just finished the experiments on fish. So we know that fish are not that important hosts as we were afraid before. And so on. So there are many questions. And no, I don't know what I wanted to say, so I'll skip it. But I know I wanted to show that we really don't know enough. I want to show you some particular situations from our research in Laos. It was last year. We did some investigation for presence of cantonensis in Laos. And this is typical Asian market. So to be very honest, if we consider that we are in the hotspot of the infection, almost anything you buy on the market can be source of the infection. So it's not that easy to study how the real infections are happening. We know that there are the cases. We know sometimes there are clusters of the cases. And I know, and this is the one that was described by our colleague Matthew, based on the case of cluster of human cases after consumption of a monitor lizard. But if we really look to the market, we don't know much. Yeah. So these are just the pictures from the real Asian market there. So what we can say that what's not important for human infection are the rats. We already know the rat is definitive host. So you can eat as many rats as you want. You can get different infections, but definitely you will not get infected by Angiostrongylus cantonensis. This we can conclude. So they are important in the life cycle, but not important in the epidemiology of human infections. If we go further through the market, the snails, sure, if they are raw or if they are being processed, then this is questionable. I was discussing this with colleagues, the local team of the colleagues from Laos, and they were almost laughing, saying, "Yes, but we don't eat the raw snails. We never eat raw snails." They even brought me an old cooking book showing that yes, the snails should be really properly boiled or properly roasted. So the risk of infection is very low, but what's not considered is the preparation of these snails, because you buy them alive or freshly dead, as you see on the pictures. We know that many of them can be infected. We know they contain the larvae, and the larvae from dying snail are actively escaping out. So basically in a dish with the snails, they can be thousands and thousands of the larvae. You bring it to your kitchen and you start cleaning it on your table, on your kitchen desk, by your knife, by your spoons, forks, or whatever. So it really creates an interface for the transmission that is not understood at all. And it can be one of the common ways how the people get infected. Maybe not with high doses of the larvae, but with small numbers of the larvae for sure. And this might explain the high seroprevalence rate, for example, in Thailand. Because if you get infected by low number of larvae, you get the antibodies, which means you are seropositive, but you don't develop the disease. And this is exactly what we see in many areas of Southeast Asia. So maybe this is the way, but it's difficult to prove, of course. Vegetables, sure. Difficult to say how frequently, but you can buy tons of raw vegetables. And if you really search them properly, sooner or later, you will find small snails in them or slugs. And if you are unlucky enough to chop it into your salad, probably this is one of the ways of the infection. What about the crustaceans? What about amphibians? What about the others? With high probability, depending on the cooking procedure. Again, there is the possibility of contamination of the kitchenware. Just doesn't mean that if you fry your frogs, it doesn't mean you don't contaminate your own kitchen and so on. And so again, huge question mark. Fish, probably only short-term infections, as I mentioned. The poultry, no idea. There were a few experiments done showing that the birds are not sustaining the infection. But this is something that is to be repeated, and this is something we are just planning to repeat in the near future. To go to the market, examine mainly the ducks, because the ducks are well known for their affinity towards eating the mollusks. And we want to start examining the ducks on the markets in Vientiane, in Laos, to see whether they can serve or not as a source of the infection of humans. So that's it for now, Cantonensis. Yeah, well-known parasite with quite fascinating opportunism in the life cycle and with still a lot of persisting question marks, including our immediate vicinity, which means including European ecosystems. So let's go a little bit to the few remaining species. We still have some time, so it's, I think, 10 minutes more and we are done, maybe 15. Angiostrongylus costaricensis. As the name says, it's a parasite distributed across Latin America. It's also zoonotic, and it's quite different from Vazorum. It's different in the location, and it's different in the pathological processes behind. So this is more or less the distribution. So it doesn't matter the color, so simply it was reported in these areas. Probably is present also in the central part of South America, just there are no data from that. So this is from a very nice and very recent review by our Costa Rican colleagues. So well done paper, so you can go through that. I'll just took a little bit from that. So the distribution is clear. It's associated with some local rodent species. It's a zoonosis. The humans get infected the same way by L3 larvae. So up to now, it's very similar to Angiostrongylus vazorum. But then the pathogenicity in humans is absolutely different. Because the larvae, they develop in the mesenterial arteries. Sorry, the larvae, they migrate to mesenterial arteries, the L3 larvae, where they continue developing into the adults. And the adults, they start laying the eggs in the mesenterial arteries, not in the pulmonary arteries, but in the mesenterial ones. And then it's quite spectacular difference between humans and the rodents. So let's start with the rodent, because this is the typical host. So what's happening in the rodents? The worms are in mesenteric arteries. It's the left side of the picture. They lay the eggs. The larvae are hatching from the eggs, and they penetrate into the intestinal lumen, being shed in the feces. Nothing spectacular, to be honest. There is mild inflammation, of course, but the rats are happily living with that. But what's different in humans is that in humans, because it's an accidental host, we can say aberrant host, where the infection is not going normally. The eggs they produce so strong eosinophilic infiltration, as you see on the picture, but also on the drawing, that basically the eggs are trapped in this inflammatory tissue, in this inflammation process. They are not hatching and they don't reach the intestinal lumen, but causing the massive inflammation in the intestine. And this is the pathology of the human infections. So the process is basically the same in the beginning, but the consequence is quite different, which is in some aspects similar to Cantonensis, because even in Cantonensis, the process in definitive host and aberrant host is the same. The larvae migrate to the brain. The only difference is that in the aberrant host, like humans, they cause massive inflammation and they don't continue further. In the normal hosts, like rats, the larvae somehow pass through the brain, reaching the lungs. So I promised I will not speak much about the Vazorum, and I'm really not going to do so. Just very briefly, the worm that you probably know well, because it's present also in Italy, typically circulating among the canines, with a huge involvement of foxes as natural hosts, like natural definitive hosts, and the mollusks and the amphibians, lizards, and probably small mammals involved as paratenic hosts. quite pathogenic infection, depending on the intensity of the infection. Many dogs are asymptomatic, but many of them can be significantly symptomatic. But what's important to say that the clinical symptoms are very, very unspecific. So to really diagnose Angiostrongylus vazorum is not easy. And what's important is, I have some slides for that maybe later, and it's important to keep it in mind that the larvoscopy is the only way that can really detect the larvae in the feces. And so far, the larvoscopy is not the most frequent method for the coproscopic examination of the dogs. Most of you have experience from parasitological lab, and you know that in majority of cases, the diagnostic laboratories perform only flotation. By flotation, you cannot detect it, we can say, or it's very difficult to detect it. So question is how much we know about the occurrence of vazorum in Europe. What's known that its occurrence is not homogeneous. There is already kind of textbook study by team of Eric Morgan from 2009, I think. So it's more than 10 years or 15 years old almost. Not almost, it's 15 years old. Based on the data from UK and Western Europe and based on some ecological modeling, they show the probability or the potential of Angiostrongylus cantonensis being distributed in Europe. And more or less, this is really true. And the situation, it's not changing dramatically since then. So the parasite is very abundant in the western part of Europe in mild, I would say, Atlantic climate. Why it is like that, we don't know, but it works. The model more or less works. Then going towards the east to more continental climate and going towards Mediterranean to more Mediterranean climate, the infection is, the prevalences are decreasing. Again, why it is like that? Because it's probably because of the snail and slug ecology. Maybe the feces are drying too fast. Maybe the larvae are not surviving that good. Difficult to say. But on the contrary, no, not on the contrary, but still, if you look to Italy, it's not really in the black color, most of that. But still, there are a series of studies from Italy clearly showing that the infection is present and proving the involvement of the wildlife in the local epidemiology or circulation of Angiostrongylus vazorum. First study on wolves. Second study I mentioned there on red foxes, and there is another one on dogs and so on. So infection is well known in Italy, and the prevalence probably depends or differ based on the local conditions, for sure, but also based on the methods you use for the detection. So if there is a laboratory which is not standardly using larvoscopy, the rate of detection would drastically decrease. Yeah, another one showing just that the spectrum of hosts can be quite broad. We are still speaking about vazorum, infection of a fennec in one of the Italian zoological gardens, but not surprisingly, fennec is a carnivore, so why not? How the infection is happening, we already discussed, and it's more and more clear that it's not by direct eating of the snails and slugs. Even though many dogs are quite stupid, including my own, they don't really eat snails. So then we should consider other ways of the infection, which is either through pathogenic hosts or through contaminated water, which I already mentioned before. And this is also the information that should be passed to the dog owners. If you are living in the endemic spot of Angiostrongylus, and this is happening, if you check the internet, it's more known among the dog owners than among the veterinarians. And for example, in those endemic areas like the Canary Islands, Australia, and so on, the owners, Hawaii, they are really aware of presence of Angiostrongylus, either vazorum or cantonensis, and they really recommend or care, sorry, they really care about the quality of the water and contact of their dogs with mollusks. So are there any other species of Angiostrongylus to be mentioned? I will mention probably only one, and it's Angiostrongylus. We can either read it Latin way, Dujardini, or from the original French, Dujardini, because it's named after the helminthologist named Dujardin. And doesn't matter. What's important that it's a species which is relatively common in some species of European rodents, in wood mouse and in how you call it, Cletrionomis, a kind of vole, forest vole, or whatever, I don't know the English name, and without any direct implications for other animals, but the infection was repeatedly reported in monkeys in the European zoological gardens. It was reported in calitrichids in northern Italy, and it was also reported in France. So in some situations, when the primates have access to mollusks that are living in the environment where the hosts live, infection can be transmitted, and these hosts can be infected. Very recently, there was a very interesting case from Switzerland by Angiostrongylus dujardini or whatever, that completed the life cycle even in a bird. It was in parrot, which is very, very unusual. Then the mammalian parasite is able to reach a maturity also in avian host. But this is the report, and it is well documented by the team of parasitologists from Bern University. So this is it. So it again shows how opportunistic these parasites are. And I wouldn't be really surprised if this species of Angiostrongylus can infect also humans, or would infect humans. Because if it is able to infect monkeys, why not humans? Maybe it's just overlooked. Maybe we don't know enough. Difficult to say. And just to make the spectrum of important hosts full, together with Crenosoma, Angiostrongylus, it's a common parasite of red pandas in zoological gardens. Again, not surprising if you imagine how the red pandas are normally exposed in very humid, green type of enclosures where the snails and slugs for sure find their way to. So no surprise that these animals that are probably eating mollusks quite a lot, not because they are hungry, but maybe because they are bored in the zoo, they get infected. And almost every group of red pandas around Europe is infected by one or more species of Metastrongyloids, and they can cause even death in them. So we are closer to the end. So if we kind of summarize the important species and important hosts of Angiostrongylus, we can say that in case of Cantonensis and Malaysiensis group, humans, but also dogs and other wildlife, are aberrant parathenic hosts or aberrant hosts, which basically suffer of the neurological symptoms of the infection. If you look to Costaricensis, then the humans are in a position of aberrant definitive host, because we already, as we mentioned, the worms are reaching the maturity in the intestine, but because of the inflammation, the larvae are not able to finish the development, but still the humans are definitive hosts, aberrant though definitive, or definitive though aberrant. And if we go to Vasorum, then the pathologies which are associated with Vasorum in dogs are pathologies in natural definitive hosts. And the pathology itself is caused by the fact that worms are blocking the pulmonary arteries, causing the insufficiencies in the circulation and the respiratory and so on, and symptoms. Yeah, so it's quite different. The pathology behind is quite different, and it's always connected to the biology. So this is why I spoke that much about the life cycles today, because only understanding the life cycles can help us to understand the epidemiology and to also start some preventive measures to stop the most risky behaviors. We cannot eliminate the parasites from our environment because it's associated, Vasorum is associated with foxes, which are everywhere. Cantonensis is associated with rats, which are everywhere. But we can modify our behavior a little bit. We can be maybe more careful when we are preparing our vegetable salads. Maybe we can stop eating the raw mollusks. Definitely we can stop eating the raw monitor lizards, and so on. The same in the dogs. Yes, we can care more about the quality of the drinking water, for example. We can replace the water frequently enough. Maybe we can even take the dishes inside overnight, so they cannot be contaminated by mollusks, and so on and so on. But to do this perfectly, or to do this efficiently, we need to understand the life cycles. And this is why, for me, Cantonensis and other Angiostrongylus are really exciting examples what we can do, what we can research or do research on, what we can understood to do so also something practically, because just by deworming it cannot be solved. None of this infection can be, or maybe in dogs, preventive anthelmintics, but infections of Costaricensis and Cantonensis, it's not matter of deworming or antiparasitic drugs. It's matter of preventive measures. Yeah. So, and this is just to remind that also very little tiny snails and slugs can be efficient hosts and source the infection. And that's it for today. And if you have any questions, please feel free to ask.
I have a question.
Yes, Mattia.
Can those larvae survive in salt water?
Probably not really. There were some experiments, and in the true like marine water, they cannot survive. Question is how it is in so-called brackish water, where the salty water is mixing with the fresh one. So they can survive longer, but in the normal marine water or ocean water, not.
Thank you.
So probably in oysters, or eating the oysters, you are safe.
Okay. Any other question? Seems you have been very, very complete, David.
Yeah, 2 hours about one worm is, I think.
Yeah, yes, you. Many thanks, and see you on 18, right?
Yeah, tomorrow, Thursday.
Okay, perfect.
Yeah. Okay, guys. So see you then, and have a nice afternoon and evening and whatever. See you soon. Bye-bye.
Thank you. Bye.
Thank you. Bye. Thank you. Bye.